afternoon. Thanks, Amy, for the wonderful introduction. Yeah, so while he's preparing slides, I just tell you guys, all the data I'm presenting today is our ongoing work. Nothing has been published, so some interpretation may be not final, but just for discussion. So I like this Vanderbilt Peabody compass, very beautiful. Some might be familiar with that. So what I'm talking about is modeling SHC sigma mutation from stem cells. Yeah, to mouse models. Yeah. Okay. Is it better? Okay, sorry. So first, as Amy mentioned, the many mutations either directly or indirectly affect GABAergic pathway has been associated with multiple genetic epilepsy. So our lab have originally studied GABA-A receptor, but right now our main focus is SLC6A1. So so brain is such organ of complexity, but just look at the smallest functional unit in the brain, the synapse. You will see how GABAergic signaling pathway is presented. So here, this is a presynaptic neuron, as Amy already talked about. So presynaptic neuron release GABA, and GABA-A receptor, what we have originally studied for many years, is like postsynaptic, and a GABA transporter, GAT1, is located either presynaptic neuron and astrocytes. So all these different components work as a symphony. They work not act alone, as I said before. So we can, maybe from therapeutic point of view, if we can leverage each component to treat the patient. So as I said, we have originally studied many years on GABA-A receptor. So not much information about the GABA transporter. However, based on our work in the last 2 years, we figured out a lot of GABA transporter actually share a lot of features, like ion channel, especially for the intracellular protein processing, the post-translational modification. Many drugs, anticonvulsant or sedation, they work through GABA-A receptor, and some GABA transporter directly. Yeah.
So you can see your slides better?
Oh yes, I feel that.
Well, you'll need them both.
Yeah, yeah. So that. Yeah. So, yeah, I said many drugs. Dr. Rogatsky is the expert. He's going to talk more about that. So I'm going to go. So we focus on SLC6A1, as the previous speaker talked about. Now, many mutations scatter around the whole protein peptide, and that's not specifically some area. However, we feel maybe from, oh gosh. So, from transmembrane domain 4 to 8, probably relatively like a hot spot. There are more mutation cluster in this area. So our in last 2 years, we have very extensively looked at 3 mutations, but we look at over 20 mutations. So right now, I'm just giving an example because Dan and Catherine talk about autism in these mutations. We have a mutation here, P361T. This is associated with epilepsy and autism. So here, ISO is in agreement with what Daniel has talked about. All these mutations seems more like inside of the protein, not much at the surface. So here, let me, should be able to play. Yeah, so you can see all this mutation more like inside of the protein. Oh no. So this is the EEG of a patient with this is a 6-year-old girl with autism and epilepsy with a P361T mutation. So here showing like a bilateral prefront slow wave, 2.5 to 3 hertz EEGs. And this is showing like a multifocal bilateral monomer discharges. And the patient also have very clear autism features. And we, in collaboration with a group in Missouri University, we use machine learning to look at over 20 mutations associated with a spectrum of epilepsy, intellectual disability, and autism. So this is I'm going to show 2 examples. So this is P361T, the patient with autism and epilepsy. So here you will see in the patient, in the mutation, they lost hydrogen bond. So this, because loss of hydrogen bond, the protein is destabilized. However, by comparing with multiple mutation, we think each mutation is a little bit different. So this mutation is S295L. So this mutation, you will see instead of loss of hydrogen bond, they can form more weak hydrogen bond. So as a result, the protein stability is a bit different. So there are many ways to study the effect of mutation. Some people use electrophysiology, some with behavior, some with stem cells. So in our lab, we have over last probably 16 years from GABA receptor to now GABA transporter. So we use different models. So first, we start from HEC cells. So we can screen many mutations, and then we kind of choose a few to more like detailed study in neurons and astrocytes. Now iPSC cells depends on which cell the mutant gene is expressed. So for SLC6A1, because it is expressed in both neuron and astrocytes, so we use both neuron and astrocytes, also include patient iPSCs. So some people in the audience asked about how this mutation behave. So we look at over 20 in the last 2 years. So this is actually I should show one in HEC cells. So right now this one is from neuron, it's a mouse cortical neuron. So we express, and now we know in mouse cortical neuron they have endogenous GAD1. So we subtract that, so compare, we normalize to the cells. We transfect it with the wild type. And from here, you have a wild type with the untreated, and 966 means the GABA inhibitor. Dr. Rodaski is very familiar with that. So and you look down the activity of GAT1, and you see what is mutation function like the inhibitor. So in short words, it's very consistent. All this mutation cause either partial or complete loss of function. And then, because we're not just look at the function, because there are many reasons, there have been many reasons to give loss of function. Some may be channel kinetics, some may be trafficking issue. So we look at the trafficking and also look at the channel itself. So first we ask how much mutant protein is at cell surface. Because traditionally we use surface biotination, which is pretty low throughput. And so we developed a high throughput with like a slow, like flow cytometry. So here we identified both methods give us very similar data. So we think that's very reliable, so we can use the flow cytometry. So this way we can look at multiple mutations, like 20 one day. So we look at this and we quantified. We found this in HEC cells, the surface definitely compared with the wild type, they have a variable degree of expression. However, compared with the wild type, all of these mutations have reduced expression. And then we wonder, where are they inside cells, right? They are not just, it's not produced. We have no idea. We just talk about loss of function. So we express them. This is live cells, by the way. This is HEC cells. So we tagged the either wild type or the mutant with YFP. So we can directly visualize and confocal. And then we co-localize with a marker, just only expressed in ER. So we know where are they. And then we use a drug, tunicamycin, which causes ER stress. So we found that the mutant actually have a very similar expression pattern like tunicamycin. And these are all live cells, by the way, right now. So this is astrocytes, because we said GAT1 is expressed in astrocytes. So look at the wild type and the mutant. So we look at multiple different mutations. So here we show two. So you will see actually each mutation is a bit different. Look at P361T and S295L. So they both can show more around nuclear region. However, the pattern is a bit different. And we found that astrocytes, because the very spreading, actually is quite nice. It's very nice to do trafficking. So this is mouse astrocytes, and we have a human GAT1. And this neuron, this is not very well done, but this is live cells, so I think it can still be useful. So we can appreciate. So the mutant and the wild type are different, and between mutant, they are also a little bit different. And this I did last year, so just a regular wild type mouse to say GAT1 in mouse tissue is more like a homogeneous distributed in cytosol like synapse. Now here, this is the stem cells, so iPSC.
Use the microphone a little more. It's hard to hear.
Can you hear me right now? Yeah, so first you will see the overlay. The second one, the OCT4 is the marker for iPSC. So that means the cells has a pluripotency. And the GAT1 is the antibody we get from SY. So we have validated, it's a very good one. And the TOPO3 is a marker for cell nuclei. So you can see at a very early stage, at the stem cells, you will see GAT1 is expressed. Also in the mutant, so first one is the corrected cell line. This is thanks to Amber and Scott. And first panel is corrected, and second is the patient one. You see the GAT1 is definitely expressed in iPSC cells. And so we also did staining, and we kind of tag the wild type or the mutant. We try to see in astrocytes, because we see in astrocytes is really expressed well. So we found definitely you see there are different pattern compared with wild type. So the mutant is quite different. So we have not done with this experiment, so I'm not make too much conclusion from that. And so some people ask about if the mutation, if the GAT1 can have dimer. So I want to say the answer probably likely yes, because look at here with this, you see at 67 is the monomer. So this is what we often see on Western, but you will see like more like a double the molecule weight. So you will see likely you will have a dimer. And then this is the iPSC cells, and then we also did a Western in iPSC. We compare with mouse tissue. So you will see the correct C represent corrected, and then PAT is a patient, and then compare with the mouse tissue, the stem cells GAT1 is expressed, but there are different patterns. So that suggests GAT1 in stem cells and GAT1 in mature tissue is subject to different post-translational protein processing. And with help from Michelle, also connected by Amber, we get the patient iPS, the fibroblast. And we have a fibroblast from ATCC. So we compare with multiple different tissues and cells. We want to see if GAT1 is expressed. Actually, I want to say the answer is yes. So from very beginning, you see from fibroblast, either normal and the patient, you will see expression of GAT1. And also in neuron and the mouse tissue, again, is consistent with our previous data. The protein processing is a little bit different. So stem cell stage and the mature stage, they are different. Before I come here, I also was able to do some GABA uptake activity in stem cells. So this is very, very low level compared with the one we expressed with a recombinant transporter. However, you will see compared with the HEC cells, the stem cells, you have a double the amount. And in the patient cells, you definitely have a reduction. It's very similar to RC96, which is a GAT inhibitor. So actually, everything is consistent here. That means the patient definitely have a reduced function. And I know some audience also wonder in astrocytes how much GAT1 is expressed. So we have some data. This is done by Sarah. We have not quantified this, but the pattern is very clear. So astrocytes, this is P0, means we just culture from mouse. Usually our experiment done with passage 2. Clearly, endogenously, astrocytes express abundant GAT1, and we can express and we can distinguish the wild type versus mutant. In astrocytes, we clearly see the mutant and the wild type, the behavior different. And also we did a neuron. We did a multiple mutation in mouse cortical neuron expression here. You will see each mutation, they have a different pattern. However, if you want to draw some conclusion, I would say majority of them have reduced, and also they have a lower, like immature protein compared with the wild type. So, and then we made a mouse model. So we made a A288V because, as Catherine said, this is a common mutation. We made actually before has been identified as a common because we look at the trafficking, so we feel this was interesting. So we made one anyway. So I found this common, so it's very good. We compare with a lot of GABA mutation mouse model. So I will show. I wonder. So I will show video. It should be able. How can I get a video here? I should be able to. Oh, yes. So left side is a white type. So basically, that's very simple. Like a retinal reflex is done by Carson, Dr. Rogaski's student. So the mutant one. So this one is before we did any genotype. So we see the mouse definitely just yeah. So it's hard to flip over. So we actually end up hype out. Yesterday I did it myself too. There are more data come out. It's very similar. So and later on we did a genotyping. We identified this is a heterozygous. Yeah. So we also did some. This is by Gerald. Did some anxiety. We use I leave the zero maze. I know Francis used the plus maze. We use the zero maze. So we don't have a lot of animal, but the data is showing a trend. The mutant, the heterozygous animal showing the trend to have anxiety. And now we did a Barnes maze. Barnes maze we compared before with the water maze. It's less stressful, so we use for cognition because the kids have an intellectual disability. I'm going to play the video. I want to play. Yeah. So basically, the mutant will spend more time to find a target hole, while the wild type, can I play this one? Oh. Come on. Come. Yeah. Oh, okay. So you can play it together. So we see the difference. Yeah. So basically, the wild type will find a target hole very quickly, while a mutant will spend much more time. So we just show. Yeah. So we're running the experiment. How many minutes I have? Yeah. So basically, we are not done with all this behavior test, and we have not done EEG. So, but we already say we already observed the heterozygous. By the way, compared with Francis homozygous, our data all from heterozygous, which is a patient condition. So we see phenotype. So we are going to finish all this and compare with probably heterozygous, and then we're going to receive S295L. So compare that. So with that, I think probably we can open a lot of discussion because I go really quickly. So if anyone have any question, and we can discuss.
Okay, questions.
Your demonstration that the GAT1 protein is not trafficked to the membrane when you have the SLC6A1 mutation is really intriguing. I mean, as I read the micrographs that you showed, it looked like there's actually potentially even more GAT1 in the astrocytes in the mutation model, but they weren't at the membrane. Do you see that same pattern in neurons?
So right now, for what I'm showing you, especially the first one, I have both astrocyte and the neuron. That's live cells. I think the data from astrocytes, I did all of that. Sarah did culture, but I did all this visualization. Is very consistent. Is very clear around the nuclei. Very clear. In neuron, because
in astrocytes, right?
Oh, this is astrocytes. So this one, astrocytes can be good model. Can be good model to study trafficking for GAT1. For neuron, because you have the dendrites, really more complicated structure. However, from I said not done yet. We definitely see the pattern is around the somatic region in the neuron, in live cells. But we did one different mutation not reported here is V511M. That one, we see some at synapse. So neuron, I don't think we have solid data yet, but I want to say from across different cell types, the trafficking machinery actually is pretty conserved. Even each cell type have different structure, but I think in general, like we look at the biochemistry, when you digest them with the endo-H and the PGSF, so you remove glycan, either those added yet ER or added in later endosome, we can tell the pattern of how this protein get processed. So if it's mutant, majority of them, the mutation, the generated protein is not mature. So you are gonna, normally we're gonna see 3 bands, like 108, 106, and 96, something like that. But with the mutant, majority are stuck at 96. So this way we use multiple, this one is because each model will give you something, but how can we get something real, right? Can boil down to something real, we can get for therapy. So this is why we use HEC cells, astrocytes, neuron, stem cells, mouse model, and we look at all of them, and so we know why this mutation gives this. So we get an explanation. So I think this is what we try to reconcile the discrimination from these models.
Well, I mean, you would imagine that the same profile would be seen in neurons. They actually express GAT1 to even higher degrees. And so if that's the case, it actually suggests a very interesting 2-part toxicity model. One, the protein doesn't get to the membrane, so you don't have uptake of GABA at the membrane level. That's a loss of function. But 2, you have what looks like an aggregate, or maybe a much higher concentration of it intracellularly, which could be like a toxic gain of function. So there may be both mechanisms going on. And if that's true, is there any evidence that GABAergic neurons themselves die at a faster rate with this mutation? Are they more vulnerable in general, so that perhaps the neurons die in addition to losing the transporter? They themselves are vulnerable.
Yeah, that's a very good question, because we have worked on this extensively. So I think right now we have a project, Sarah, actually she studied cellular stress for different mutations, not GAT1, but GABA. We have compared, we definitely say. So, yeah. So we right now we already include GAT mutation. So we don't have a poster yet, but we now poster about the GABA receptor. But I think this is the unexplored area. So when we talk about mutation, I think we must consider 3 areas. So most of the time, everyone think, oh, loss of function? Yes, that's first. Second, if a mutation is loss of function, how this mutation interact with the remaining wild type, that we must take into consideration. And thirdly, how this mutant protein interact with host cells. So either neuron or astrocyte, based on whatever cells they express, how these host cells respond, that one has to be addressed. I think the whole field, we do not understand this area. So that's why I think when you look at why these heterozygous knockouts have a very little phenotype. We look at not just the GAT1, we have looked at knockout, but look at the GABA receptor, we paired multiple mutations. Knockout, you have basically very little phenotype, but the lock-in, same missense mutation, and you have a sewed up, you have seizures, you have everything. So what's going on there? So I think that area, so I think these 3 things we have to take into consideration. The response, the host cell's response, that part is almost, we have 0 information from there. So right now we start to add on like a GAT mutation to our cell stress panel. So in addition to loss of function, or if they have dominant negative effect or not, and how cells, the host cells, either astrocytes or neuron, respond to a mutant protein. So that part we are learning.
Okay.
Yeah.
Thank you so much, Katty.